What is fermentation and how does it work? What is fermentation? Fermentation is the metabolic process where microorganisms consume carbohydrates like glucose or starch to produce alcohol or acid. It was initially developed to stabilize and preserve perishable food, but is now also used to further improve the nutritional and functional traits of certain foods. Examples of fermented foods are cheese, beer and wine, kimchi. What are the two main types of fermentation? There are two main types of fermentation: alcoholic and lactic acid fermentation. Alcoholic fermentation, also called ethanol fermentation, is a type of fermentation which products are ethanol and carbon dioxide. It is performed by yeast species, specifically Saccharomyces, for the production of alcoholic drinks such as beer and wine, as well as foods such as bread. Lactic acid fermentation breaks down sugars, producing energy in the form of ADP. This process is so named because lactic acid is a byproduct of this reaction. A typical example of lactic acid fermentation is making yogurt by the bacteria Lactobacillus. What is the process of fermentation? The fermentation process starts with glucose, a simple sugar that undergoes glycolysis to produce pyruvate. Pyruvate will then be converted to ethanol with the use of yeast for alcoholic fermentation, or lactic acid with the use of lactic acid bacteria for lactic acid fermentation. How to perform a successful fermentation. From employing the appropriate working space during sample preparation and fermentation procedures to storing the involved microorganisms properly, fermentation requires ideal working conditions and environment. Here are the laboratory equipment that are needed to set the fermentation up for success. Cultivation and storage of microorganism. ESCO isotherm 4-stand natural convection. Laboratory incubators are used to culture Saccharomyces at 30 to 35 degrees Celsius. Lactobacillus at 37 degrees Celsius. These laboratory incubators are crucial for maintaining the optimum temperature and condition for the microorganisms to flourish. ESCO refrigerators and freezers are used to store Saccharomyces at -20 or -80 degrees Celsius. Lactobacillus at 4 degrees Celsius. A -80 degrees Celsius lab freezer is the best storage equipment for the pure liquid culture of yeast, while probiotic bacteria strains can be kept at 4 degrees Celsius in culture. Sample preparation and aseptic techniques. ESCO laminar flow cabinets are used for safe handling of microorganisms and avoid cross-contamination. It is designed to facilitate the aseptic transfer and smear of probiotic bacteria and yeasts onto solid media. Fermentation is a well-known natural process used by humanity for thousands of years. It is not only useful in food manufacturing, but also encompasses the production of desirable products, such as the generation of fuel from vegetable sources and more. And as the world progresses for future discoveries in the science fermentation, so are the technologies. Find out more about the ESCO Life Sciences products through the links provided in the description below.